Hello, everybody. Welcome back. So, in our last lecture, we were talking specifically about an introduction to the polymerase chain reaction, which is basically a way to harness the power of DNA replication in a laboratory environment. And so that's going to be the focus of today's lecture, is really understanding how scientists and specifically forensic scientists use this method to understand and create a DNA profile. So again, PCR is essentially mimicking DNA replication. So think back to what we were looking at previously, and what was the first step that happens in DNA replication? And that's the breaking of these bonds between the adenine, thymine, guanine, cytosine. So breaking these specific bonds has to be the first step. So how do we do that in a laboratory environment? We actually just use the simple power of heat. So enough heat helps separate the DNA strands so that we have one side and then we have this other side. But then what we want to do is allow them to come back to not necessarily come back together, but bring in the additional things that we have in this test tube, which include each of these individual base pairs. And that when we actually cool this reaction, so we first heat it up and then we start to cool it, the extra base pairs that are floating around will go and bind to these different nucleotides. And because we have our DNA polymerase added, essentially the reaction is able to knit together these two sides of the DNA, and just like that, we have replicated a DNA strand in a test tube using basically what we know and understand from the cellular process. So let's zoom out and understand sort of what that looks like. So initially, in the start of our PCR reaction, we added to the test tube the original strand of DNA, excess nucleotides of adenine, thymine, guanine, and cytosine. We added the DNA polymerase, that important enzyme, again, ACE for enzyme. And then lastly, primers that allow us to specifically look at a specific section of DNA. So in our previous slide, we looked essentially only at this first step, where we were able to create an identical strand of DNA from this region. But what we really want to do is we want to replicate that many, many times so that we're able to have lots of repeated sections of this original DNA. And this step is called amplification. And really, it allows us to make billions of copies of DNA, even just starting out from this very tiny sample. So even though we might only have a couple skin cells, for example, or a single hair, in the laboratory, we're able to amplify the genetic structure of that single hair to many, many copies, so we can very definitively say this is what we have in our sample. So this process brings us to the idea of DNA profiling. And basically, DNA profiling is just taking advantage of the fact that no two people have the exact same DNA sequence. And we know that because we're able to do something called whole genome wide studies that basically looks at one complete set of genetic instructions encoded in the DNA of an organism. So it takes and it looks at every single base pair that an organism has. And we've been able to sort of compare across many, many people that no two people have the same complete set of genetic instructions encoded. However, this is a very time-consuming process, although it's getting faster and faster every day. So whole genome sequencing is certainly something that has even tripled or quadrupled in the space of time needed to create a whole genome. But it still is a complicated process, and certainly not in the capabilities of law enforcement or forensics. So what do we do then? We can't create this whole genome sequencing for every single person because it's going to be very time consuming and very expensive. So what we do is we take use of a shortcut where we're able to use, again, this polymerase chain reaction to amplify only specific segments of DNA. And these short segments of DNA that we're going to be talking about are very important regions called short tandem repeats, or STR. So what specifically is a short tandem repeat? Essentially, they're just sequences of the chromosome which DNA sequences are repeated. So for example here that your book gives, adenine, guanine, cytosine, thymine may be repeated over and over again in a chromosome. So we have here one chromosome where we have the maternal chromosome and the paternal chromosome, and we have this area of short tandem repeats. And we can see that this is repeated 6 times in the maternal chromosomes and 4 times in the paternal chromosomes. And importantly, these short tandem repeats are in the same places along chromosomes. So we can look at one person, another person, and then a third person and know that there's going to be a short tandem repeat at that area. However, the length of those short tandem repeats is going to vary throughout the different populations. And because this exact length of these short tandem repeats vary from person to person, we're actually able to utilize this information to create a genetic profile without sequencing the entire genome of a person. We're able to focus in on these short tandem repeats. So using these short tandem repeats is what allows us to make a DNA profile. And that's what we're going to sort of pick up in our next lecture, is understanding how we use the polymerase chain reaction, PCR, to create a DNA profile using STRs, short tandem repeats. Okay, we'll pick up there, and I will see you soon.